All right, let's continue our lecture. And today we'll talk about two more topics. The first one we'll talk about is the diagnosis and treatment of viral infection. So when we learn bacteriology, we learned a lot on the diagnosis of bacterial infection. So usually we follow the standard procedure and we learn how to collect the specific specimens. And after collect the specimens, how we analyze the bacterial morphology and how we culture them and how we test their biochemical properties so that we can derive useful information for diagnosis. Well, for viral infection, the diagnosis is a little bit different because viruses are quite different from bacteria. You cannot easily culture them on petri dishes, and they require some live cell to replicate. So it's not easy to culture. And the other way is that viral particles are so small, you cannot observe viral particles under light microscope directly. So yeah, today that's what we will learn today. So the first thing for diagnosis of viral infection, and you can see that the procedures here are somehow different from what we learned before. You can use different ways, different methods for diagnosis. First one is that if you have a patient coming into your clinic, you can check the signs and symptoms of your patient. And usually, according to your knowledge and according to your experience, based on the symptoms, probably you can guess what type of infection, what type of virus it is. For example, this is the herpes simplex type 1 virus infection. And you can also collect the specimen and apply the virus to cell culture. And on the cell culture, you can see the morphology of the cells change. So you know that that's the effect of viral infection. And you can also culture the cell and then inoculate the virus to the cell culture or the embryo so that you can check the change there. Or if you really want to check the morphology of the viral particle, you can use electron microscope to see the particle. Or we can use molecular biology methods to detect the viral nucleic acid, either DNA or RNA. So you can use like PCR to amplify the viral DNA or RNA, or you can use some probe to hybrid with the viral nucleic acid. So that's the molecular biology method. Or you can detect the viral antigens with Western blot. So that's a method using antibody to detect the existence of certain viral antigen, viral protein. So these are different ways we can use for diagnosis. And the first thing first, we need to collect specimen from your patient. We need to decide where to collect the specimen. After you see the specific disease symptoms, you can decide the location, the part of the body to collect a specimen, whether it's a blood, whether it's some infection site fluid or something else. And once you see the onset of disease, once you find the symptoms, we should collect the specimen as early as possible. You don't want to wait till it is too late, because our immune system is trying to handle the viral infection. If you collect the specimens too late, probably you won't get nothing from it. You will get nothing from it. And we should keep the specimens fresh. And we should place them on ice so that the viral particle won't be degraded by enzymes. And if there is some other microorganism like bacteria, and low temperature on ice can inhibit the bacterial replication. And to further inhibit any bacterial replication in the specimen, we can also add some antibiotics to the specimen that can inhibit the bacterial growth, or it can kill the bacteria. So that's the key point to collect specimens of viral infection. Then we want examine the virus. You can use microscopy and staining to visualize the viral particle. And we mentioned that viral particles are so small, you cannot use a light microscope to see a single virus. You need to use electron microscope to see the morphology of virus. The problem is that electron microscope is not everywhere. This equipment is relatively expensive. It costs a lot of money to buy one. In our university, in Zhengzhou University, we only have several electron microscope here. And most of the hospitals or labs, they don't even have electron microscope. Another way is to check the existence of virus using immunofluorescence staining. You can use some antibody to recognize the existence of viral antigen. And that antibody is conjugated with a molecule which can give off fluorescence. So that if you see the fluorescence, you will know there is such viral antigen. Well, definitely, you can only see the light signal. You cannot see the viral morphology because it's too small in light microscope, even with a fluorescence light microscope. We can also culture the virus and further analyze its properties. We can use cell culture. And when you culture the virus, you can check whether the virus causes change on the cell, whether there is some cytopathic effect, whether there is viral interference, whether there is production of hemagglutinin, and etc. We can also culture the virus in chicken embryo. For example, for the influenza virus, we usually culture them in the chicken embryo to harvest large number of viruses. Or you can inoculate the virus into animals so that you can amplify the virus in some animal. So these are different types of viral culture. And we can detect viral antigens, for example, some protein from the virus. And the way to detect the antigen, usually we use immunoassays, different types of immunoassays. Immunoassay means that we detect something using some antibody. We produce some pure antibody, some monoclonal antibody. That antibody can recognize certain viral antigen specifically, so that based on the antigen-antibody reaction, we can design different types of immunoassays. For example, the immunofluorescence, like we talked about just now, that's one of them. We can also use the enzyme-linked immunoassays. For example, ELISA. So that's enzyme-linked immunoassay to detect some viral proteins. Or previously, people used a radioimmunoassay. So this is the method you incorporate radioactive material to your antibody. And after binding to the antigen, you can detect the radiation signal. So this is a an old way and classical way. But now, because radiation usually it's not safe to technicians, we are trying to avoid any radio immunoassay. But enzyme immunoassay and immunofluorescence are very popular now. Or we can detect the nucleic acid of the virus, either DNA or RNA. We can use PCR to detect the existence of nucleic acid. Right now, the popular PCR people are using is the RT-PCR. That's a real-time PCR. So this is a real-time PCR from Bio-Rad. It's a famous brand. And usually the signal looks like this. So if at the beginning there is a lag phase, no increase, then suddenly there is an increase. This increase means that there is some amplification, there is some nucleic acid in it. And during this special time, during the pandemic, now almost in every country, we are using real-time PCR to detect the existence of coronavirus RNA. So it's very popular and it's widely used. And now in China, the big hospitals usually they have one of these machines, but in small hospitals they usually don't have one. I heard that recently in China, the government want to equip small hospitals to have this machine so that they can have a quick detection, quick diagnosis at the local site. They don't have to send the specimen or send the specimen to other labs. That will save a lot of time. Okay, the next method is to detect viral antibody. And as you know that the viral antibody is not produced by viruses, it's produced by the infected host. So you collect some blood or some serum from the host. If there is virus infection, you can find the specific antibody in the host. And of course, you can carry out the experiment in different ways. For example, you can use viral antigen and mix with the serum and see whether there is a neutralization. So that's a neutralization test. Or you can use the anti-serum to carry out the hemagglutination inhibition test. Or you can do the complement fixation test. So in different ways, you can decide whether there is neutralizing antibody, there is antibody against the virus in the serum. So this is another way to decide to diagnose virus infection. Okay, the next we'll talk a little bit about the antiviral therapy. Well, unlike bacterial infection, which we can use different antibiotics to inhibit bacteria, for a lot of virus infection, that we don't have specific medicine to inhibit the virus replication. So that's bad. And we don't have any antibiotic to treat against virus because they are working on bacteria. So generally, the antiviral therapy is kind of limited right now. Even though there are some methods we can use, like antiviral chemotherapy, there is some chemical medicine you can give to your patient. So that can somehow inhibit the viral replication. Or we can also use interferons to inhibit viral infection. And the best way and most efficient way is to use viral vaccines. And that cost is much lower than medicine. And if there is vaccine available, try to get one, because vaccine is much powerful than the medicine. So that's different ways for viral therapy. Okay, let's check the chemotherapy methods. We can use either nucleoside or nucleotide analogs to interfere the viral nucleic acid synthesis. We can use reverse transcriptase inhibitor to inhibit the reverse transcription, like for RNA, for HIV. HIV is a retrovirus. We can use these reverse transcriptase inhibitors, or we can use protease inhibitors or other types of antiviral agents. And with the next several slides, we'll introduce some examples here. Oh, this picture shows us different ways, different strategies we can take to inhibit HIV infection. There is a lot of information in these slides. You can check it later. But now we'll talk about several examples on chemotherapy of virus infection. The first one is that we can use nucleoside or nucleotide analog. You know, this is the synthesis of RNA molecules. And this nucleotide, the normal one, contains this OH group on the single ones. And let's see from the beginning. This OH group, it can be incorporated into this RNA chain. This is a DNA chain, sorry. This is a retrovirus, for example, HIV. But if you use some analog, this analog is modified here. This is not an OH group anymore. This is N3, containing 3 nitrogen atoms. And let's see from this is a normal one, normal nucleotide. And you have one nucleotide analog coming in, this N3 coming in. Now it blocks further extension of the chain. So it blocks the synthesis of DNA molecule. So it blocks the HIV infection. This is a nucleoside or nucleotide analog, the first one. The second one is we can use reverse transcriptase inhibitors. This is also designed for HIV infection. You know, that's a retrovirus. It needs to reverse transcribe its RNA to DNA. But if we have some inhibitor to inhibit the viral RNA to form DNA molecule, we can inhibit the viral infection. This is the second mechanism. Another one is protease inhibitor. I don't know whether you are familiar with viral behavior. And if you check the viral genome, including the coronavirus, you'll find something interesting because in their genome, the virus, because it's so small, it doesn't want to waste any energy, any material. It just links its DNA, its genes, one by one, head to tail. So if you check the genome of the virus, you will find that all the genes, or most of the genes, are linked together to form a long fragment. And when this fragment is transcribed or translated, they will be transcribed and translated together. So finally, it will form a giant peptide. So that giant peptide contains several proteins in it. But it needs a protease to cut the peptide into pieces so that each piece is a functional protein, like here. The protease come in to cut the peptide into small pieces. And only these small pieces are functional protein. But if you block the activity of the protease, like this, using inhibitor, the protease cannot cut the giant peptide anymore. You cannot generate functional proteins. So this way you can inhibit the viral infection. You can inhibit the viral spreading. So this is another mechanism using protease inhibitors and also interferons. And we learned that there are different types of interferons, and type 1 interferon, including interferon alpha and beta, and they can carry out the antivirus activity. And this picture shows us how the interferons interfere the viral infection. So it happens this way. This is a cell, a host cell, which is infected by a virus. And this virus can be sensed by the cell. So the cell will produce some interferon molecule. Well, this interferon molecule doesn't influence this infected cell. It's secreted. It is secreted, and the secreted interferon will influence the neighboring cell. This is a neighboring host cell. And it binds to a receptor and tells the neighboring host cell that, well, be careful, some virus is coming. So that this neighboring cell will start to transcribe some gene and produce the antiviral proteins. And these antiviral proteins will prevent infection by the virus. So this is the way of interferon to inhibit viral infection. Okay, so this is the first topic we want to talk about, the diagnosis and prevention and therapy of viral infection. Do we have extra time? No. Well, those videos you can also watch them online. We don't have to play them here.